Good evening, everyone. So today we will discuss about amino acids. So actually in the syllabus we have proteins. So before going into the topic of proteins, first we should know about amino acids. So I'm discussing about amino acids. So amino acids are the monomer units. Okay, they are the monomers which form long polypeptide chains of proteins. That means if the chain of amino acid is very long, we call them as proteins. If it is short, we call them as polypeptides simply. So we have L and D amino acids in nature. So usually most of the amino acids which are involved in the metabolism, they are L amino acids. Okay, and their derivatives. So what is this L and D amino acids means? They are, of course, they are mirror images of each other. There are two configurations of amino acids, and also it is based on their rotation. Okay, so when we pass an plain polarized light through this amino acid solution or powder, so then the light will turn into left or right direction. So based on that also we will classify. Okay, so mostly L amino acids, they participate in cellular functions like in nerve transmission, then in the synthesis of porphyrins, purines, pyrimidines, which are nucleic acids, that means which are the bases involved in nucleic acids like adenine, guanine, cytosine, thymine, uracil, then urea. So in their synthesis, usually we see this amino acid. Then short polymers of amino acids, if present, they are called peptides. We cannot call them as proteins, but we call them as peptides. Okay. So these peptides are involved in various roles like neuroendocrine system. That means both in nervous system and endocrine system. So in the endocrine system, mainly the system deals with the hormones. So hormones, they regulate various metabolic functions, right? We all know that various processes which are important processes are modulated using hormones and also hormone releasing factors. Then in the nervous system, when we see like a signal has to pass through the body. So first the signal goes to the neurons, and from neurons the signal may go to another neuron or another muscle cell. That we call them as,
Excuse me, ma'am.
yeah.
Ma'am, which chapter, ma'am?
So same I think fourth unit or so.
Fourth
You
unit.
just you just check the syllabus code.
Okay,
Yeah.
fine.
Yeah. Okay. So when the signal has to pass from neuron to another neuron or neuron to a muscle cell, so there will be an action potential and a signal transmission that takes place. So this is modulated by neurotransmitters. They are small chemicals, there's acetylcholine, all these things. So they will mediate this neurotransmission. So these neuromodulators, neurotransmitters, hormones, hormone releasing factors, all these could be peptides. Peptides can act as all these components. So when we see the general structure of amino acids, so except proline, all other amino acids will have similar structure. So what is the structure means? To a center carbon atom, we have 4 different functional groups which are linked. That is one side hydrogen, other side amino group, other side carboxylic acid group, and we have R chain. R chain means side chain. So this usually R chain differs between different amino acids. So that means we have different types of side chains in different functional groups, which will be altered. Okay. So in glycine, we see like 2 hydrogen atoms only in the glycine. R group is also a hydrogen atom. Okay, so that is the only difference. And here, usually normal pH, physiological pH, the carboxylate ion, that is COOH, will be negatively charged. That means COO minus. So in that state, it will be existing. Whereas this amino group is protonated. That means it will mean NH3 plus form. Okay. And these amino acids can be classified based on 4 different ways. So into 4 different groups. So one is based on their structure. Second is based on side chain characters. Third is side chain based on nutritional requirement and metabolic fate. So based on the structure. So when we say structure, mostly the other 3 groups like amino group, carboxylic acid group and hydrogen, they remain same. Only thing is R group will differ. So based on the R group or functional group, so we will get different structures. So the amino acids are classified as aliphatic amino acids. So in the aliphatic amino acids also, we have monoamino monocarboxylic acids. That means now when I shown you the structure of amino acid, you have one amino group and one carboxylic acid group. So that means monoamino monocarboxylic acid groups. Okay. So such amino acids which are having only one amino and one carboxylic group are called monoamino monocarboxylic acids. So examples are, first is simple amino acid, that means glycine and alanine. They are very simple, like only single group will be present, like hydrogen or like that. Okay. Then branched chain amino acid. That means at the R group, if we have side chains which are branched in nature, then they are called branched chain amino acid. So examples are valine, leucine, isoleucine. Then OH containing amino acid. So if in the R group, we have OH group containing, OH group is there, then they are called as OH group containing amino acid, like example serine 309. Then sulfur containing amino acids, which are cysteine and methionine actually. And when 2 molecules of cysteines, because they have sulfur. So in one cysteine we have a sulfur group, other also we have sulfur group. When 2 cysteine molecules and their sulfur groups are forming a disulfide linkage, then that compound is called as cysteine. Then amide group containing amino acids. So that means in place of R side chain, if we have amide group, then they are called as amide group containing amino acids. Examples, glutamine and asparagine. Then we have monoamino dicarboxylic acids. So till now, whatever the amino acids we have discussed, they are monoamino monocarboxylic acids. Now, where from the other carboxylic group is coming? It is in the R side chain. Okay, so that means in the backbone of the amino acid, we already have one carboxylic acid. Again, in the R group also, if we have carboxylic acid group, then we call them as monoamino dicarboxylic acid. Okay, examples are aspartic acid and glutamic acid. So these two are derived from the like asparagine and glutamine. Okay. Then next is dibasic monocarboxylic acid or diamino monocarboxylic acid. Here, that means in the backbone structure, we have one amino group, one carboxylic acid group. And in the R group also, if we have amino group, then diamino monocarboxylic acid or dibasic monocarboxylic acids. So examples, arginine and lysine. Then aromatic amino acids. So till now, whatever the amino acids we have discussed, they are aliphatic amino acids. Now aromatic amino acids means we have a ringed structure. Okay, so tyrosine, phenylalanine, and tryptophan is also an aromatic. And again, heterocyclic amino acids also we can call them, because many different ring forms, if they are present together in the R group, then we call them as heterocyclic aromatic amino acids. So they are tryptophan and histidine. Amino acid is example proline. Then we have derived amino acids. So derived means usually the amino acids which are present, they are alpha amino acids. So what is alpha amino acid? I will just show you the structure once again. So this is the general structure. So here if you see, so the first carbon in this compound is COOH. Okay, that is the functional group, carboxylic acid group. The carbon which is present, that is the first carbon. So the adjacent carbon attached to the first carbon is called as alpha carbon. Suppose if there is another carbon adjacent to that, then we call it as beta carbon. Then gamma, delta, epsilon, like that, we will have numbering. Okay. Yeah. So as mostly the functional groups are present on the alpha carbon atom, we call them as L-alpha amino acids. But there may be derivatives of these amino acids which are non-alpha amino acid. So they are beta-alanine, gamma-amino butyric acid, that is called GABA, delta-amino levulinic acid. Then derived and incorporated in tissue proteins. That means some amino acids are derived from these standard amino acids, and then they are incorporated into the tissues also. So example, hydroxyproline, hydroxylysine. That means the hydroxyproline is derived from proline, hydroxylysine is derived from lysine. Then derived but not incorporated in tissue protein means some amino acids, they are derived from the amino acids but they are not incorporated into the tissues. So they function separately, like ornithine, citrulline, homocysteine, arginosuccinic acid. These are some of the examples. Then the second type of classification is based on the side chain characteristics. That means how these side chains are behaving, like R group. So first classification is amino acids with non-polar side chains. So non-polar means they cannot dissolve in water. Okay. So examples: alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, and proline. So here, why they cannot interact with water? Because they cannot give off protons and they cannot react with the protons. Okay, therefore they cannot participate in hydrogen bonds or ionic bonds. And these side chains are thought to be oily because they are not water soluble. Hence, we call them as oily or lipid like amino acids. Okay, and they promote hydrophobic interactions rather than hydrophilic interactions. Okay, then next group are, they are polar but uncharged side chains. So example: glycine, serine, threonine, tyrosine, cysteine, aspargine, and glutamine. So here the R groups are polar in nature. That means they can interact with water, but they are uncharged. They are uncharged. So there will be no charge. They will be neutral in nature. Okay. And cysteine and tyrosine, they remove, means they can lose proton at alkaline pH. So they will form charge, negative charge, but at alkaline pH, but not in the neutral pH. Then serine, threonine, tyrosine, they can form hydrogen bond because of presence of hydroxyl OH group. And aspargine glutamine, they have carbonyl group, extra carbonyl group, carbonyl group and amide group. So they can participate in the hydrogen bonds. Then next is amino acids with charged side chains. Means either positive or negatively charged side chain will be present. So amino acid with positively charged side chain are called basic amino acid. Examples: lysine, arginine and histidine. Whereas amino acids with negatively charged side chains are called acidic amino acids, and examples are glutamic acid and aspartic acid. These are hydrophilic in nature because as they have charge and they have capacity to interact with water, they will be hydrophilic in nature. Okay. Then classification based on nutritional requirements. So that means how much they are required as a nutrition, through nutrition. Okay. So in the previous class, when I was discussing about fatty acids, I told you about essential fatty acids, non-essential fatty acids. So in the same way here also we have essential amino acids, semi-essential amino acids and non-essential amino acids. So essential amino acids means which cannot be synthesized in the body and they should be supplemented through diet. Sorry, you can't see the screen?
Yes, ma'am. We can't see the screen.
Nobody can see?
No, ma'am.
Okay, let others respond. Only,
Yes, ma'am. They are messaging me that there is no screen.
No, ma'am, we can't see the screen.
okay, means can you see the slide or it is not moving or you cannot see the screen
No,
itself?
no, ma'am.
It
It's
is
black.
totally
It's
blank.
black. Nothing is visible. It's totally
Okay,
black.
okay, I'll share again.
Ma'am, this is about respiration, ma'am.
No, it is about amino acid.
No, actually, which unit? Because I'm not having textbook. That's why.
See, according to your syllabus only I'm telling, like you have physiology and biochemistry, no?
Yeah.
Yes, ma'am.
Yesterday you have explained all about lipids, no, ma'am.
Yes,
Today you are explaining about amino acids, right?
yes, right. Now can you
Okay,
see?
ma'am. Yeah, ma'am. It's
Yes,
visible.
ma'am. It's visible.
Okay. Yeah, okay. Just a second.
It's blinking.
What? I'm just changing the slides. Wait.
Okay.
Okay. Can you see now?
Yeah,
Yes,
ma'am.
ma'am.
Yeah, yes, ma'am, it's visible.
Classification
Yes,
based on
ma'am.
nutritional requirements, right?
Yeah,
Yeah?
ma'am. Okay.
Okay.
Yeah, ma'am. Okay.
Okay, fine. So now essential amino acids means the amino acids which we have to provide through diet because they cannot be synthesized in the body. Okay. So examples are valine, isoleucine, leucine, lysine, methionine, threonine, tryptophan, and phenylalanine. So all these amino acids we have to supplement through diet. If they are missing in the diet, then they can cause some kind of diseases or disorders within the body. Whereas semi-essential amino acids, they can be synthesized in the body, but not in the appropriate amounts. Means very low quantities we can means they can synthesize. But during some conditions, our body requires more amount of nutrients. Like if we see in pregnancy or during some growth of children, okay, in babies, then if a person is a patient, like some transplantation or due to some diseased condition, there should be a repair of the tissue or something. Then what happens? They require more amount of a particular nutrient. So at that point of time, though they are synthesized in the body, we should also supplement it through diet. Okay. So such kind of amino acids are arginine and histidine. The non-essential amino acids means those amino acids are usually synthesized in the body in the appropriate amounts, and even though they are not supplemented in the diet, they cannot cause any disorders or diseases like glycine, alanine, and others. Okay, and means other remaining amino acids what are not listed in the above list. Okay. Then next is based on the metabolic fate. So that means, see, during metabolism, so metabolism means what? Catabolism and anabolism. So anabolism means synthesis, catabolism means breakdown. So breakdown, first what happens? Any amino acid, when it undergoes catabolic process, so whatever the structure I have told you, amino acid, that will be broken down into individual atoms. Okay, like carbons will be separated, hydrogens will be separated, then nitrogens will be separated, like that. Okay, they will form a pool. Then from that carbons, nitrogen, hydrogen pools and all these things, they will be utilized for the synthesis of some other compounds like urea or purines, pyrimidines, carbohydrates, etc. So during this process, like how this carbon amino acid skeleton is utilized, in which process it is utilized. So whether it is involved in the glucose synthesis or whether they are involved in the production of ketone bodies. So usually see when we fast, what happens? Our ketone bodies will increase in the urine sample, right? So that ketone bodies are also synthesized. So based on the process in which these amino acids are involved, we can classify them as glucogenic and ketogenic. So some amino acids can form both glucose as well as ketone bodies. They are called both glucogenic, and ketogenic, like isoleucine, tyrosine, phenylalanine, and tryptophan. Then some are purely ketogenic in nature, okay, like leucine, lysine. Some are purely glucogenic, like the other 14 amino acids, like glycine, alanine, valine, all these other 14 amino acids, they are purely glucogenic. That means they are involved in glucose production. Okay. So this is the classification of amino acids. Then I told you about standard amino acids. So there are 20 standard amino acids. So standard amino acids means we have many amino acids existing in nature. But if you take any protein from any living organism, whether it is plant, animal, human, or microorganism, any protein, you can see only 20 amino acids which are arranged in different sequence and different combinations. So that's why we name those 20 amino acids as standard amino acids. So now, although we have about 300 naturally occurring amino acids, as these 20 are commonly seen, they are called as primary or standard amino acids. Then 21st amino acid is selenocysteine, and the other commonly seen ones are pyroglutamate and pyrolysine. Now, how we give the naming to an amino acid? So usually, how we have to represent an amino acid means first we have to write full form. Okay, either we can write the full form like glycine, alanine, valine, like that we can write, or you can give a symbol. There is a code which is given. So that is, suppose here example cysteine. First three letters like CYS. This is a 3-letter code. We can represent like that also. Histidine HIS, alanine ALA, like that we can give. Or the next one is unique first letter code. That is cysteine means C, capital C, histidine means H. Like that we have to give. So usually when we are in the text form, when we are writing about a single amino acid, we can represent as cysteine, full form. Okay, but when we look for the sequence of a protein, you cannot write everything. So usually we'll have a single letter code. Now during this process, we will have some discrepancies. Like what happens? See, alanine is there. You can give A. But again, aspargine is there. That also starts with A. Aspartic acid is there. That is also starting with A. So how to follow the rules? Okay. So first, next, if suppose many amino acids are having the same starting letter, then we should give priority to commonly occurring amino acids. That means, suppose alanine, aspartate, aspargine. So, which is a commonly occurring amino acid? Alanine. So, you give letter A to the alanine. Okay. Then glycine, glutamate, like that we have no glutamine. So, everything is starting with G. So, as glycine is commonly occurring, we give G to glycine. So preference is given to the commonly occurring amino acid. Then next is based on the sound. Suppose you see tryptophan, tyrosine. Okay, these two are also having T. They are starting with the T. So how to give the name? So we can give based on the sound, similar sound. Tryptophan means R, usually it sounds similar to W. Okay, so you can give W for that. Then phenylalanine, Fa. Okay, the sound Fa means F. So phenylalanine and proline, we have both starting with P. So as they are starting with sound, F is given. Then next choice is letters close to the initial letter. Suppose you have aspartate, it should start with A, but already A is given to alanine. So which letter is close to A? B. So B is given. Then lysine, LYS, L was given to leucine. So you can give K, which is close to L. That is another form. So like this, already the coding is given to all the 20 standard amino acids. So for the exam point of view, not only this means your annual exams, but for competitive exams, like if you are preparing for entrance exams or for PhD entrance or CSR or any other exams, always you have to remember these 20 amino acids. Okay, they are very, very important. You have to remember the names, codes, what are the functional groups, because all the three groups are same. Because in the amino acid structure, you can see amino group, carboxyl group, and hydrogen group. Only the R group is different. So that R group you have to remember. Okay, and the classification, nutrition means which are essential, which are non-essential, and which are amino acid containing, which are acidic containing, which are basic amino acids, which are aromatic, aliphatic, like that you have to remember. So this is very important topic where you can expect many questions. Okay, and the special groups present in amino acids. So rather than normal groups like hydrogen, OH group, or like that, methyl group, you have some special groups which are present in the amino acids. Like for arginine, you have guanidinium group. Phenylalanine, you have benzene group. Tyrosine, you have phenol group. Tryptophan, indole group. Histidine, imidazole group. And proline, pyrrolidine group. So these are the special groups you can see only in the amino acid side chains. And this proline is having a secondary amino group also. That's why we call it as imino group. Okay, amino acid rather than amino acid. Though it is an amino acid, we call it as amino acid. So now what are the properties of amino acid? So this classification is very, very important for your future, any examination. So you have to practice the structures, remember the groups, okay, names, everything. Then properties, they are colorless, crystalline in nature. Some are sweet in nature like glycine, alanine, valine. Some may be tasteless like leucine, and some may give bitter taste like arginine, isoleucine. So when they have some property which is unique like sweetness, we can use them for commercial purpose. Like aspartame, which is an artificial sweetener, is prepared using aspartic acid and phenylalanine. Okay. Then these are soluble in water, acid, alkalis, but not in organic solids. And they have high melting point. That means only at temperature more than 200 degrees centigrade, they can actually melt. Then we have an important concept which is called isoelectric point. So this isoelectric point is nothing but usually the amino acids when they are present in the solution form, they exist as Zwitter ions. Zwitter ions is nothing but they don't have any pH. They don't have any charge. They will be neutral in charge. So that means the pH at which they don't have any charge, they will be neutral in nature. Such pH or such point is called isoelectric pH or isoelectric point. Okay, so that means every amino acid will have a different pH where it exists as without any net charge. Okay, that is called isoelectric point. So in acidic medium, what happens? It will bear a positive charge and they exist as cation, and in the alkaline medium they exist as anions. So we will see one example. So here you can see at pH 1, that means acidic pH, what happened? So NH, the amino group is in the form of NH3 plus and carboxylic group is COOH. Now the net charge is positive charge plus 1. When the pH is raised to 7, that is neutral pH, a proton is removed from COOH. And now you have NH3 plus COO minus. One positive, one negative. So it will become neutral. So this is zwitterion. That means at pH 7, this particular amino acid is existing as zwitterion. And when the pH is further raised to alkaline pH, then a proton from NH3 plus is removed and it will become NH2. Now you have the net negative charge, that is anionic form. So now for this amino acid, the isoelectric point is 7, pH 7. Okay. So the same thing is explained here. So here, titration, how we calculate the isoelect already calculated, means we will have a standard chart of isoelectric points for all the 20 standard amino acid. But we should know, suppose we have discovered some new amino acid, then we have to do all these things, right? So how we calculate this isoelectric pH or isoelectric point means we have to use this formula pK1 plus pK2 by 2. So pK1 is nothing but where it is forming from cationic form to neutral. Then pK2 is from neutral to anionic form. Okay, so it will form pK1 plus pK2 by 2. Okay, by calculating this, we'll get the isoelectric point. Then optical properties of amino acids. So optical properties are nothing but as this alpha carbon atom is bound to 4 different functional groups, we call it as chiral carbon or optically active carbon atom. Only except glycine, because glycine will have 2 hydrogen atoms linked. So that's why it is not optically active. And these amino acids have 2 different forms. They are D and L forms, which are mirror images. Mirror images means the structure is same, but instead of left, the other will be right. So as our hand, if we see in the mirror, how it appears, left fingers to the right, the thumb especially. So in the same way, mirror images are formed. They are called enantiomers. And usually in proteins, we see the L configuration. D configuration is seen only in some antibiotics and bacterial cell walls. Okay. Now, reactions of amino acids. So I won't discuss them in detail because in your syllabus you don't have that depth. Okay. But I'll just give the glimpse of the reaction. So amino group can get involved in some types of reactions. Then carboxyl group are involved in some reactions. Side chains are involved in some reactions. And both amino and carboxyl groups are involved in some reactions. Excuse me. So amino group is mainly involved in oxidative deamination. That means deamination means removal of amine group. Okay. Then transamination means transfer of amine group. So in these two reactions, usually this amino group will participate. And also carbamino compound formation. So amino group with carbon dioxide, when reacts, it will form carbamino compound. Then we have reactions due to carboxyl group, which are decarboxylation. That means removal of carboxyl group, then formation of amide linkage. Please mute your microphone. The next side chains are involved in ester formation, ester bond formation. And also the SH groups, that is sulfur groups, as I told you, cysteine. So cysteine containing sulfur group. So if two cysteine molecules, they form disulfide bonds, they will form cysteine. Okay. Then transmethylation. So in the methionine, the methyl group is transferred from one compound to other. So that is called transmethylation. So these reactions are important for the metabolism to form derivatives. But if we keep on discussing like that, only the reactions will take a lot of time. So that depth is not required. So I'm skipping that section. Then the important reaction here is reaction due to both amino and carboxyl group. So here, this is very much important because it is the basic reaction for the formation of protein. Here, how peptide bond is formed means, see, in the first amino acid, you have carboxyl group COOH, and in the second amino acid, you have NH3 amine group, NH2. So this H from amine group and OH from carboxyl group, when they react and the water molecule is removed, and the bond which is formed is called as peptide bond. Okay, so in this way, the proteins are formed. Like first amino acid is forming peptide bond with the second amino acid. Second amino acid is forming a peptide bond with the third amino acid. Like that, it keep on continuing to form a protein. Okay. So the special functions of amino acids are usually they are incorporated in the tissue proteins. And from tryptophan, niacin, serotonin, melatonin are synthesized. From tyrosine, melanin pigment, thyroid hormone, catecholamines are synthesized. From glutamic acid, gamma aminobutyric acid, which is a neurotransmitter, is synthesized. Then from arginine, nitric oxide, which is a muscle relaxant. Then we have heme, creatine, then glutathione, porphyrins, purines, pyrimidines. So all these are synthesized from the amino acids. So they are derivatives of the amino acids. And to check like which, if you have given a sample of amino acid and asked you to identify which amino acid it is, we have a set of reactions using which we can actually identify what is the amino acid present. So we have different tests like ninhydrin reaction for alpha amino acids, xanthoprotic test for aromatic amino acids, Millen's test for tyrosine, Biuret test for free amino acids. Like that, we have many tests, okay, using which we can practically identify what is the amino acid. Okay, so this is all about amino acids, like classification of amino acids, what are the different 20 amino acids and what are their functions. Now, how these amino acids are involved in the protein synthesis. Okay, that we will discuss. I will share another PPT. Yeah. So now, as I told you, through peptide bond, different amino acids, they bond together, they form a chain and they form a protein. So once a protein is formed, it will immediately fold into a three-dimensional conformation, which is very specific. Okay, it is not random. Somewhere it is folding in some form. No, it is very specific to each protein. So when I discussed about enzymes, I told you like their structure depends on their function depends on their structure, because when how they fold will determine what is the function. Okay, so the folding is very specific, and the fold is called as native fold. That means each protein will have a native fold. It folds only in a particular pattern or format. So why it is folding only in that format? Because of favorable interactions. So when they are folding, they are not loosely attached. Okay, so there will be interactions within the folds. So that interaction should be favorable. Only favorable interactions are allowed. Okay, and the free energy changes are also matters. Okay, so what are the different favorable interactions involved in proteins? Are hydrophobic effects. Okay, as some may not interact with the water. Then hydrogen bonds, then London dispersions. So there are some weak attractions within the proteins. Then electrostatic interactions between charged groups. They form salt bridges. So like that we have different interactions participate in the protein folding. So we have four levels of protein structure. One is primary, secondary, tertiary, and quaternary. So in the primary structure, if you see, the three-letter code of the amino acids are given in a sequence. Like the amino acids are arranged in that order. Proline, alanine, aspartic acid, then lysine, threonine, aspargine, valine, lysine, alanine, tryptophan, glycine, lysine, valine, like that. So these amino acids, they form into different structures like either sheets or coils or turns, okay, or helix. If they are helix, then they are called alpha helix. Once they are called secondary structures. So once the secondary structures are formed, different secondary structures together, they can form tertiary structure. And these tertiary structures are the actual protein structure. They are called polypeptide chains. And these multiple polypeptides, they can form a quaternary structure. So what is a primary structure? Here the peptide bond is formed. It is the structure of the protein which is partially dictated by the peptide bond. So here the peptide bond will have a double bond, hence there will be resonance. So you might be knowing that what is resonance. So when there is a double bond, the bond will shift into the adjacent molecules, like adjacent atoms. Like here you can see C double bond O. That double bond is shifted to in between carbon and nitrogen, right? So that is called as resonance. And based on this resonance, usually the bonds present between carbon-carbon and carbon-nitrogen, we will have different angles. So the bond between carbon-carbon, though it is a single bond, so it is given with the name psi. Okay, it's a symbol psi. And between carbon-nitrogen, it is phi. So based on the angle, we have to note what is the angle of psi and what is the angle of phi. And when we plot these values in a standard plot, which is called Ramachandran plot, we will get to know if we have the sequence of amino acids, we can determine whether that forms an alpha helix or beta sheet. Okay. So there are two common arrangements in the secondary structures. They are called alpha helix and beta sheets. So alpha helix are stabilized by hydrogen bonds, and beta sheets are also stabilized by hydrogen bonds in the adjacent segment. Sometimes they can form a random coil. And now no need to remember all these spacing and all these things. I'm just skipping. So usually it is right-handed helix. So you can know the difference between left-handed and right-handed helix from this image. So how this the stability of this helix can be determined. So based on the amino acids present, we can actually say whether the alpha helix is stable or not. That means if alanine and leucine residues are more in the sequence, they form alpha helix. But if proline or glycine are present, they will break the helix, hence they cannot form the helix. They can form beta sheet. Then the dipole moment also varies based on the amino acids present. And here the CO, carbon oxygen, that bond, the carbonyl group, the carbonyl group present that gives a negative dipole moment, whereas amide group gives a positive dipole moment. Okay. Then beta sheets. So beta sheets are nothing but they are arranged in the form of a sheet, but not the helix. Okay. And they can be of two types, parallel and anti-parallel beta sheets. So parallel beta sheets means if the two sheets are in the same direction, then we call them as parallel beta sheets. If the two sheets are in the opposite direction, then they are called anti-parallel sheets. So like this, these are parallel, all in the same direction. This is anti-parallel. One is moving to the one direction, and the second one is in the opposite direction. Then beta turns means some amino acids, some chains, they can form turns. Means here if we have proline in second position and glycine in third position, then usually we can expect a beta turn instead of beta sheet or alpha helix. Then here isomers, proline isomers. So most of the peptide bonds which have trans configuration, they don't have proline. While if they have cis configuration, then they are involving proline and they can form beta turns. Okay, that means these two different types are called proline isomers. Okay. Then comes the tertiary structure. In the secondary structure, just remember that there are alpha helix, beta sheets. In beta sheets, they are parallel, anti-parallel, and they can also form beta turns. So that's enough. Then in the tertiary structure is nothing but the overall spatial arrangement. There will be weak interactions between the amino acids, that hydrophobic and polar interactions, and sometimes disulfide bonds are also stabilized. And here there are two types of tertiary structures and two types of proteins, fibrous and globular. So when I discussed about the enzymes, I told you about that they are globular proteins, right? So fibrous proteins are examples, alpha keratin, silk fibrin, and collagen. So alpha keratin, which is present in the hair, nails, feathers, they are all fibrous proteins. They are structural, okay, mostly. And keratin consists of alpha helix majorly, silk fibrin, that is silk threads. So they are made up of beta conformation, and collagen has triple helix, not two helical structures, but triple helix, which are present in the tendons and bone matrix. So in the hair, we have keratin. So it has disulfide bonds. Okay. So when we have a straight hair, some people will have straight hair. That means they have disulfide bonds in the keratin. So what happens? Some people want to make it curls. So there is a treatment. Okay. So for that, first what they will do is they will break all these disulfide bonds so that the hair will become curly. Okay. Again, if they form disulfide bonds, it will become straight. So like whatever the cosmetic treatment we will undergo, like for straightening of hair or curling of hair. So the basic mechanism is breaking or bonding of disulfide bonds. Okay. Then collagen. So collagen mainly present in the connective tissues like tendons, cartilages, bones, cornea. They are rich in glycine and proline. Hence, they will be usually left-handed helix. And sometimes they also have another helix that is right-handed. So it will form a triple helix structure. Like this, you can see. So here in collagen, we can also see a 4-hydroxyproline. So here the post-translational processing is done by prolyl hydroxylase, which is an enzyme. It requires alpha ketoglutarate, molecular oxygen, and vitamin C. Okay. So these are the microscopic structure or microscopic image of collagen fibers. And these globular proteins, the other ones, they are water soluble, like hemoglobin, myoglobin, enzymes. These are all globular proteins. Then motifs. Motifs are nothing but the small folds which are arranged in a specific secondary structure. That means they may be all alpha helix or beta sheets or a combination of alpha helix or beta sheets. So these motifs are the recurring structures. That means usually in the globular proteins, you will have small motifs. That means if the protein is about 100 amino acids, some 5 amino acids will form a motif. Like that, we can have many motifs within the same protein. So this is the motifs examples: beta barrel, beta alpha, beta loop, like small secondary structures which are formed. Then finally, the quaternary structure. So it is an assembly of individual polypeptides and form a large cluster. For example, hemoglobin, you have two alpha subunits, two beta subunits. That means the polypeptides, again, they are together forming a quaternary structure. Then, once the structure is formed, again, its stability and folding depends on many factors. So the protein's function depends on its three-dimensional structure. And its structural integrity, if it is lost, then it is called as denaturation. And why this is lost? If it lost its structure, then it will lose its function. Okay. So how it is lost? It is mainly contributed by heat or cold, that is extreme temperatures or extreme freezing, or pH changes, then exposure to organic solvents and catastrophic agents like urea, vanadium hydrochloride, like that. Whenever we treat a protein with these chemicals, they will lose their structure and function. And again, after some proteins, when we remove such conditions, again they will form its native structure, which is called as renaturation. Okay. Then comes protein misfolding. So folding is also very important. So once these proteins are synthesized by the ribosomes, they will be folded and they will be transported to wherever they have to function. Okay. So when this folding is not correct, so it will cause some diseases. That is, misfolding diseases will be there, like mad cow disease and some neurological diseases within human beings also. So in brain, they'll form different misfolded proteins, and these misfolded proteins, they get accumulated and they can cause some neurological disorders. Okay. So this is all about amino acids and proteins classification and arrangement. So in the next class, we will discuss about their metabolism and also carbohydrates. Then further we'll continue. If you have any doubts, you can just ask. Okay, if you don't have any questions, you can just go through the text. If yeah,
Excuse me, ma'am.
yeah.
Some important questions about fifth semester, ma'am, about all these units.
Yeah, once I complete the topics, I'll give you. Okay.
Okay, ma'am. And
Yeah.
what about the generic, ma'am?
Sorry.
We have generic syllabus also, ma'am, in zoology, like aquaculture and sericulture.
That I don't know. You can ask in the department. I am dealing with only this paper.
Okay.
Okay.
Thank you, ma'am.
Yeah. Okay. Thank you so much.
Hello, everyone listening. Add me on group.
Send your number.
99.
1 minute.
Okay.
1 minute. 1 minute.
Okay.
995
5923
23
96
96
22.
22. 1 minute. 99523 5622
59.
59. Okay. Name?
9959239622.
Okay. Name?
Aisha Begum.
Okay.
Okay, thank you.
Welcome.